morning, dear students. Today we are going to continue the balance of transcription. In the last class, we have learned about the splicing, which are the different types of proteins, RNA involved in that splicing, spliceosome, everything. So today we are starting with the evolutionary implications of split genes and RNA splicing. How this RNA or regarding that RNAs that is doing that splicing activity. What regarding its evolution? That's what we are going to see now. First, we need to understand about RNA world. What do you mean by RNA world? Later, in the perusal of science, it has discovered about ribozyme. Ribozyme. What is ribozyme? No, ribozyme means any RNA that is doing catalyzing activity is called a ribozyme. Okay. So after the discovery of this ribozyme, scientists think that it is RNA that plays dual role. Dual role means in ancient times it is RNA itself act as genetic material and also that itself catalyze all the chemical reactions. Understand? In ancient time, there is no DNA or protein for doing this distinguished functions or different functions like to carry genetic material or to catalyze chemical reactions. For those different actions in the ancient time and the primitive time, there is no DNA or protein. That's the belief. And when that belief happened after the discovery of ribozyme. And when the scientists found about the splicing and spliceosome, then they think that it is a proof of what? Proof of legacy of that ancient RNA world. That means in the primitive times, there is no DNA or protein, only RNA. RNA is doing everything. It acts as the genetic material as well as it is catalyzing chemical reaction. That is called RNA world. So we believe. Okay. Next, we are going to see about origin of this pre-mRNA introns. In pre-mRNA, we have just seen that in our last section, last video about so many introns, how its splicing is taking place. So from where? What about its origin? How it originated? Okay, from it is thinking that from group 2 introns. About group 2 introns also we have learned in the former sections. From the group 2 introns, actually the introns of pre-mRNA evolved. That is the recent finding. It's the last finding. And regarding that, we are going to see. First, there are some endosymbion organelles. It is believed that mitochondria is also an endosymbion. Endosymbion means in primitive condition they are prokaryotes and somehow it is engulfed by some eukaryotic cell, and as a result of that engulfment it become a part of that eukaryotic cell. That type of organelles are called endosymbion. So first, in primitive state, inside the endosymbiont organelles present that group 2 introns. And those endosymbiont organelles are present inside primitive eukaryotic cell. Okay. And in that eukaryote cell present a nucleus also. And as time passed by, as a result of evolution, what happened next? The group 2 introns from that endosymbiont organelle move to that nucleus. Okay. After moving to the nucleus, what it then do? It take a permanent residence inside the nucleus where inside that genetic material. Okay, here we can see that this is group 2 introns, and we know that is the gene sequence itself. So from that sequence, it is possible to transcribe an RNA. Okay, and later as time passed by in the course of evolution, that RNA becomes snRNA. Okay, uh, from where that snRNA formed? snRNA is formed from that intron which get translocated from endosymbion organelle to nucleus. And also one more thing, this intron can change its position from one place to another in this genome because of a special property called transposition. Okay, that's also need to keep in our mind. Okay, from that sequence, what is forming? An RNA. That RNA become later in as a result of evolution, it becomes snRNA, and we have learned that this snRNA is carrying out the splicing. So from that intron that are having the self-splicing property, that property is taken over by and the RNA. RNA means the RNA that get transcribed out from this group 2 introns. Okay. And snRNA is formed. After its formation, it lost its self-splicing capability. And later this snRNA get associated with so many proteins, and this snRNA become snRNP. And this is about the origin of our present form of introns in the pre-mRNA. Okay. Next, I am going to explain about the origin of SNORNA. What is SNORNA? They are small nucleolar RNA. Where we heard about this name? Yes. We learned about this type of RNA during the processing of rRNA, ribosomal RNA. For the processing of ribosomal RNA, the main agent is this SNO RNA. How this SNO RNA or from where it is originating? That I am going to tell you. There are some genes. Genes means these genes are actually involved in the encoding of polypeptides, that is involved in the organization or assembly of the ribosomes. That genes. In that genes present introns, and during its transcription from the introns, you know what get transcribed? Some RNA, and that RNA get modified or processed into snRNA. Okay, and till now we believe or we studied that introns are simply discarded out from the primary transcript. Okay, but here in the case of these genes means the genes that is involved in the transcription of polypeptide that is required for ribosome assembly. In that type of genes, introns have a significant role. Significant role means those RNA that get transcribed out from these introns get modified into snRNA. Okay. That's about the origin. And in some type of genes, the significance or the role of introns and exons get interchange. Interchange means in that type of genes, simply suppose this is another type of gene, and in this also introns and exons are there. In this type of gene, when it is get transcribed, what happen? You know, transcribe uh, in that in that primary transcript. In that primary transcript, what all will present exons, present introns, present, and from that primary transcript, instead of replacing introns or instead of discarding introns, what is discarding and degrading? Exons are degrading. In such type of genes, there is no role for exons. Only significance is for introns. And it's introns then modified or further processed into SNO RNA. Okay. Next is about exon shuffling. Shuffling means mixing up, and exon also gets shuffled. What is the role of this shuffling? Okay. If we consider two proteins and analyze the polypeptide, sorry, the amino acid sequence of those two proteins, we can see homologous regions in those two proteins. What's the reason? One reason is exon shuffling. What is exon shuffling? Exon shuffling means regions of exons is moving between in different genes. How that movement is occurring? That movement is occurring with the help of these introns. The regions of introns breaks are happening, and but specific sequence of gene is moving to a different gene, and that may lead to the generation of new protein, and the generation of new proteins within a single generation may lead to evolution. Okay, that is the significance of exon shuffling. Okay, next is about RNA interference. Interference means hindering something. So that hindrance is done by double-stranded RNA here. What this double-stranded RNA can do? This double-stranded RNA, when it is added to a solution that contain many mRNA, it can specifically distract one mRNA that carries the same sequence as that of this double-stranded RNA. By taking this principle, we can study the role of a particular enzyme in a particular cell. How? By adding this double-stranded RNA to that cell, and as a result, when that particular gene get transcribed, and as a result of transcription, we know that mRNA, corresponding mRNA will form, and as this mRNA has the same sequence as that of that added double-stranded RNA, it can distract this mRNA, and we can learn the effect of the lack of this enzyme in that particular cell. Okay. With this, we can finish today's class and section. Thank you.